recording. And the first thing is, are there any questions? Okay, so Chapter 7 deals with the basic principles of biology, DNA replication, transcription, translation. And so this is material that you should have seen in Biology 1406. What does this topic get any easier? It should get easier this time. It really should. If you've seen it before, it is always easier the second time around. You'll see, especially because it will come back. The things that you've seen will probably come back. And so, yeah, it'll get easier, trust me. So essentially what we're going to do is we'll look at how DNA replicates itself and how proteins are made. And really, you're going to say, well, what does this have to do with microbiology? What it has to do with it is this is the way microorganisms work. And a lot of what we know about DNA and protein synthesis, we learned it through bacteria. And you'll see why. We'll talk about why is it that we learned it through bacteria. Basically because they're simpler than a eukaryotic cell would be. And the premise of it is, the idea is, if this happens in a bacteria, then should it happen also in a eukaryotic cell like a human cell? And the answer is yes, with the difference that in a bacteria, it is easier to understand because you're not dealing with organelles and you're dealing with chromosomes. Bacteria only have one circular piece of DNA, so it's a lot simpler. So nowadays, because we manipulate bacteria genetically, not only in order to identify it, but also in order to get it to do things for us, molecular and DNA biology and protein biology of bacteria are very, very important. So when you speak of a genome, that will be the entire genetic material of an organism. For a bacteria, that's going to include the DNA molecule, which is you could say the chromosome of the bacteria. It's only one piece of DNA, but it also means plasmids, which are circular pieces of DNA independent of the chromosomal DNA. So that would be all of the DNA of the cell. Gene is the functional unit of the genome. Genetics is the study of genes. Genomics is the analysis of nucleotide sequences. So we're going to start with going over the structure of DNA, which we kind of did that in chapter 2. So again, we're going to go back to DNA. This is a huge molecule. It's made of monomers of smaller molecules called nucleotides. And so many nucleotides put together is what makes a DNA molecule. And a nucleotide, in turn, is made of a sugar, a 5-carbon sugar. Attached to the 5-carbon sugar is a phosphate group. And we number the sugars. There's 5 of them. So the last one, 5' sugar, is where the phosphate group is attached. Out of the first sugar, the C1 sugar, a nitrogenous base is attached. And a nitrogenous base is a molecule that has rings of carbons with nitrogens in them also. So here's an example of a nucleotide. This molecule here is the sugar, is a 5-carbon sugar. Remember that each corner represents a carbon, so there's carbon 1, and we start counting them from the oxygen clockwise. So this is carbon 1, carbon 2, carbon 3, carbon 4, and then outside of the ring is carbon 5 right here. So attached to carbon 5 is the phosphate group. Attached to carbon 1 is the nitrogenous base, and this is a nitrogenous base called adenine. Again, a ring of carbons. There's a carbon here, and a carbon here, a carbon here, and then the rest are nitrogens. So there's your nucleotide. The sugar in the case of DNA is a deoxyribose. That's what the D in DNA stands for. Okay. Yeah, this is all bonded covalently. So this is a stable component. It's a stable molecule. These nucleotides are very stable. So we said that DNA is put together by many of these nucleotides joined to each other. And the way they're going to join to each other is the phosphate group from one nucleotide is going to bind covalently, again, a strong bond, chemical bond, to the carbon number 3 of another nucleotide. And so we have the phosphate sugar, phosphate sugar. So we call this phosphate sugar, phosphate sugar, phosphate sugar. And we call this the backbone of the molecule because this is all covalent bonds, which makes these very, very stable structures. So this is the sugar phosphate backbone of the DNA. So you probably know that DNA is a double-stranded molecule. And because the backbone is just the scaffold that's holding the bases together, oftentimes it is just depicted as a ribbon. So this ribbon is the backbone. So the ribbon will be the phosphate sugar part of the molecule. So we have DNA is a double-stranded. So we have two of these DNA molecules joined together. So we have this is the phosphate backbone, sugar backbone of one of the molecules. I'm going to put it like that. So here's one of them. The other one is this one right here. And so they intertwine with each other in such a way that the bases are facing each other. And the bases from the nucleotides of one of the strands phase with the bases of the nucleotides of the other strand. And the bases will form bonds with each other. Now, notice that these bonds are not drawn as a continuous line, but as a dotted line, which indicates weak bonds. So these are going to be weak hydrogen bonds, which are the weakest of all chemical bonds. What that means is that these bonds that are holding one strand with the other one, with the opposite strand, can be easily broken. And then the two molecules can be, the two strands can be pulled apart. Notice that here it says TGA, GTCA. Those are the different types of nitrogenous bases that we can find in DNA. So we're basically going to have a T nitrogenous base, a G nitrogenous base, A nitrogenous base, and C nitrogenous base. And notice that the T nitrogenous base pairs with the A, and the G base pairs with the C. So this is how the double-stranded molecule is put together. Notice something else. Notice that one of the ribbons, the last out of the sugar phosphate group, the last group that is free, not attached, because that's the end of the molecule, is the 5' right here. If you follow that molecule at the other end, the last group that is not attached to anything is the 3'. On the opposite molecule, the last carbon that is not attached to anything is this 3' carbon, okay? And at the opposite end, right here, 3', at the opposite end, it will be the 5' carbon. What that means, take a look at these two ribbons here. What it means is that for the nitrogenous bases to face each other, the two ribbons cannot be running on the same direction, because if they were running in the same direction, the other ribbon, the bases will be right here. So what has to happen is that the second strand has to be opposite from the first strand, and you can see here how the sugar is upside down, if you will. And if you turn it around opposite, now the bases are facing each other, and they can interact. So we call that, we describe that as having anti-parallel strands. We'll go ahead and touch on all these again here in just a second. Okay, so that's just going over the structure. So this right here is one nucleotide. This is another nucleotide. This is another nucleotide. When DNA is being synthesized, and this will be an anabolic process because we're building something. So it's an anabolic process that requires energy. So when DNA is being synthesized, one nucleotide, the phosphate group from the 5' group of one nucleotide, is attaching to the 3' carbon of the other nucleotide. So there you have the sugar, the phosphate sugar bond. Again, one end of the molecule ends with, in this case, with the 5' carbon as the last carbon unattached. At the other end is the 3' carbon that will be unattached. So this will be 5' end, this will be the 3' end. Yeah, yes, this is where thymine dimers happen, exactly. So if there are two thymines, these are adenine and adenine. So if there are two thymines adjacent to each other, they're going to covalently bond to each other, which distorts the DNA molecule. So yes, that is exactly what would happen. Yeah, that we'll see that in chapter 8. So if you have, let's see, an A and a T and a T and a C and a G, so that means the other, they're called anti-parallel strands, correct? Yeah, but we're going to mention that in another PowerPoint. I'm kind of covering a lot at the same time, going ahead of everything, but we will definitely mention it again. So this would be T, A, G, C. Okay, so here we have two T's together. And what we're talking about is something that happens by exposure to UV light. So UV light creates a covalent bond between any T's that are close together. And the problem with that, you're going to say, well, what's the big deal about that? Well, the problem with that is when that happens, when these two T's bond together, the DNA is going to distort. And it's going to, because these two T's come closer together, they need more room. And so the molecule creates a bubble to try to fit in those two T's that are now joined together. So now you have this bubble in the DNA, which will create mutations when the DNA replicates. So now we have these two T's joined together and there is a bubble there. So yeah, that's called thymine dimers. And it's the specific damage that UV light causes on DNA. So the nitrogenous bases of DNA, you definitely need to know these, are thymine, cytosine, guanine, and adenine. For practical purposes, we call them T's, C's, G's, and A's. Turns out two of them are pyrimidines, two of them are purines. Purines have two nitrogenous rings. Pyrimidines has one nitrogenous ring. So purines are G's and A's. Pyrimidines are T's and C's. So that's what they look like. And when they base pair with each other, always a purine base pairs with a pyrimidine. And turns out that guanines base pair with cytosines, adenines with thymine. When guanine and cytosines base pair, they create 3 hydrogen bonds between them, whereas adenine and thymine base pair with 2 hydrogen bonds. So that means that an AT bond could be easier to break up because there's only 2 hydrogen bonds versus GC, which are 3 hydrogen bonds, so it will be a little bit harder to break up. Remember from chapter 2 that we said RNA does not have thymine. Instead, it has uracil as its base. And uracil could base pair with adenine, just like thymine can. So they're very similar. But DNA does not have uracil. Only RNA has uracil. Thymine is DNA only. Okay, so we do know now that a adenine always base pairs with a T. So A's go with T. I guess this is just pretty A and T only. Okay, so this base pairing is what we call complementary. A's and T's going together, G's and C's going together is a complementary base pair. So they complement each other. Okay, so here's another depiction of a double-stranded DNA molecule. You can see AT, GC, TA, AT, CGs, GC. So you should know anytime that there's an A, that at the other end, the other molecule will have been base pair with it with a T. G's goes with C's, T's with A's, A's with T's, T's with G's, G with C. So this is the complementary characteristic of DNA, of the nitrogenous bases. The antiparallel characteristic of the DNA is when on one strand, the last carbon that is unattached to another nucleotide, the last free carbon, is the 5'. Notice the 3' is still part of the sugar phosphate bond. At the other end, the last carbon is going to be the 3'. And then the opposite strand, notice the sugars are going to have to be, the whole molecule is turned around so that now the last carbon that is unpaired is the opposite from the other molecule. It's going to be the 3', and at the other end it will be the 5'. So the molecules run in opposite directions. If one runs 5' to 3', the other one will run 3' to 5'. Okay, so they're antiparallel. So you should know what we mean by complementary base pairs and antiparallel strands. You should also know where is DNA found in prokaryotes and in eukaryotes. So in prokaryotic cells, DNA is found as one single molecule, which is circular. Okay, I don't have circular anywhere in here, but it's circular. Plus, prokaryotes also have plasmids, which are non-essential, small, circular molecules that are independent of the chromosome, carry their own information, and typically only for their own replication, and carry one or two more genes. So here's a plasmid, here's another plasmid, and this mesh here, blue mesh, that's the actual DNA, the chromosomal DNA. It is a circle, it's a huge, huge circle compared to the size of the cell, and so doesn't look like a circle from the picture, but it is a circle. And by the way, do plasmids work like mitochondrial DNA? Yes, they are very similar. Very, very. As a matter of fact, I would say mitochondrial DNA is very similar to bacterial DNA. So yes, they're definitely mitochondrial DNA is very close to prokaryotic DNA in more ways than one, as we're going to see. So absolutely, yes, yeah. We'll see also how plasmids are very flexible. They're drawn here as independent, but they could join with the chromosomal DNA, and then they could be pulled apart again. So they're very plastic as to what they can join with each other. Okay, DNA molecule replicates. It does so is starting at one point, which is called the origin of replication, or OR. So the molecule has one OR. In this case, the OR is right there in that little dot. And so when it begins to replicate at the end of the replication process, there will be two cells, each with their little OR there. Remember, bacteria replicate by binary fission. And better than this picture right here is the one down here. That is showing you how DNA is beginning to separate the new strand from the OR, and eventually wind up with two molecules. Plasmids, again, plasmids are not essential. So we could remove all the plasmids from a bacterial cell, and that's not going to kill the cell. Plasmids have genes that give the bacteria survival advantages. So for example, we have plasmids called fertility factors, which allows the bacteria to exchange DNA with other bacteria. There are other plasmids that have been called resistance factors because they are the ones that carry the genes for antibiotic resistance. So what is the difference between binary fission and mitosis? They both end up with true. The difference is with clone daughter cells, correct? Yeah, they both end up with identical daughter cells. The difference is that mitosis goes through a series of steps. The prophase, the metaphase, anaphase, and telophase, which we do not see in bacteria. So all of that dance of chromosomes that we see in the eukaryotic mitosis is not seen in here. The replication just involves one chromosome, which begins to copy itself, and then we have two chromosomes, and then the cell divides. So it is not so much of this movement of chromosomes and that we see. It's a much simpler process. It's also it's a faster process also because it's much simpler too. Obviously, there's a lot less DNA in a bacteria than it would be in a human cell, for example. So, the purpose of plasmids, as we identify plasmids, we give them, we identify them as fertility factors or resistant factors, depending on the genes that they're carrying. Bacteriocydins carry genes for killing other bacteria. Virulence plasmids may carry genes for perhaps toxins. Okay. So plasmids can be pretty important, especially medically. A lot of the resistance to antibiotics is going to be carried in plasmids. So this is an E. coli cell, and they've ruptured the cell, and the DNA has been spilled all over, and you can see here the plasmid. So a couple of things about this picture that is striking to me is how huge the DNA molecule is. So you're looking at these, and we're telling you this is a circle, but unless somebody tells you this is a circle, you'll never know that it's a circle. And how huge the molecule is compared to the size of the cell. So what that tells you is that humongous molecule, which is easily 100 times the size of the cell, in order to fit inside the cell, it has to be packaged with proteins and with what we call coiled and supercoiled. And this is true of eukaryotic chromosomes also. It's not just bacterial chromosomes. Let's see. So nowhere we find DNA in a prokaryote. No, they don't have histones. No, good point. Prokaryotic DNA does not have histones. Histones are the proteins that package DNA in eukaryotic cells. However, they have other types of packaging molecules. There's no way anything that big is not packaged. So there are other types of proteins that package DNA. They're not histones, but they're some other kind of protein. So yes, absolutely. Yeah, that's another characteristic, that they don't have histones. So in the eukaryotic, we find DNA in the nucleus. We typically have more than one chromosome, and the molecule is linear instead of circular. When the molecule is not dividing, it looks like a mesh of DNA. It's not until the cell enters the division stages that we begin to see the matter of fact, in prophase is when we actually see the discrete, distinguishable chromosomes. So, we're going to talk about also the other place where we find DNA in a eukaryotic cell is going to be in the mitochondria. So mitochondria have their own DNA. It's called mitochondrial DNA. And mitochondrial DNA is circular, just like prokaryotic DNA is. Okay. So it's very, very similar to prokaryotic DNA. Eukaryotic DNA has many origins of replication. Prokaryotic DNA has one origin of replication. So prokaryotic DNA has a circle and one origin of replication. Eukaryotic DNA is linear, and it has many origins of replication. And from that point of replication, when DNA is being replicated, these replication bubbles appear, and they all eventually join each other to create the new DNA molecules. So here's the DNA being packaged by these green circles, which are the histones, proteins that are going to package eukaryotic DNA. In eukaryotes, extranuclear DNA is found in the mitochondria and in the chloroplasts of photosynthetic organisms. This DNA is circular, resembles chromosomal prokaryotes, codes for a little bit of genes, just 5% of RNA and proteins. Some fungi and protozoans will also carry plasmids. So plasmids are unique to bacteria, fungi, and protozoans. Okay, so that's giving me the little mitochondrial DNA. All right, so you should be able to compare and contrast the genus of prokaryotes and eukaryotes. The significance of mitochondrial DNA being similar to prokaryotic DNA is that the theory is that mitochondria at one point was a prokaryotic cell. And the prokaryotic cell back at thousands of years, millions of years ago, was engulfed by a bigger cell and became a organelle. The other implication is because anything that there's so mitochondrial DNA is similar to prokaryotic DNA, any antimicrobial drug that harms prokaryotic DNA may also harm mitochondrial DNA. Okay, because they're so similar. Okay, you should be able to define what we mean by antiparallel strands, complementary strands. Given one DNA strand, be able to write the orientation and base sequence of the other strand. So if you're given, let's say, 5 prime, A, G, C, T, T, 3 prime, that's one strand, you should be able to write the other strand. So we know they're antiparallel, so the other strand will be 3 prime. We know they're complementary. Where there's an A, there's a T. Where there's a G, there's a C. C, G, T, A, T, A, 5 prime. So one is 5 prime to 3 prime, the other one is 3 prime to 5 prime, and the bases are complementary to each other. So you should be able to write the other strand from the example right here. So expect to do something like that for the test, for example. Okay, so now we're going to look at DNA replication. And this right here is what's happening at the origin of replication. So at the origin of replication, all the enzymes that are involved in replicating the DNA are congregated. And DNA is going to be fed through that OR. And as it goes through the OR, it will be replicated. So oftentimes it's compared to the film, the reel of a film that as it goes through it, it's coming off on the other side and it's coming out double replicated. Okay, so again, just to review what we mean by complementary, antiparallel. All right, so DNA replication creates a new DNA strand from the parental strands from the nucleotides. It is an anabolic reaction, which means it's going to require energy. Requires energy and requires building blocks, monomers. So those are the two things that DNA replication requires, since it is an anabolic reaction. The beauty of the process is that both requirements can be filled by nucleotides. So nucleotides act as both the building blocks of the new DNA molecules plus the energy molecule. So what the cell is going to use is an adenine with 3 phosphates, a thymine with 3 phosphates, a guanine with 3 phosphates, and a cytosine with 3 phosphates. So each one of these are high energy molecules that can be used to make a new DNA molecule, plus they carry their own energy. Okay, so DNA strands are complementary, not identical. So given one sequence, you could create the other. So remember that DNA is a double-stranded molecule. A double molecule is always going to be energy molecules. Yes, absolutely. A triphosphate molecule always going to be energy because that last phosphate is a high energy phosphate. So yes, they're definitely energy molecules. Yeah, so not just, and it doesn't have to be ATP, an energy molecule. It can be also GTP, CTP, and TTP. They're all high energy molecules. So when you have the DNA molecule, you have 2 strands. Each one of these strands serves as a template to create the new strand. So as you separate these strands, we're going to call these the templates. And the new strand is going to be created using the templates as the guide. So let's say this template right here will allow us to create 5 prime T A C G 3 prime, and the other template, the molecule created, will be 3 prime A, run out of room here, T G C 5 prime, and now we have 2 double-stranded molecules, black with purple, green with black, which they're both going to separate one into a different cell. So that is look at that, I forgot I had done that. So that is the trick of DNA replication, is the fact that the DNA can be separated, and each of the strands is going to serve as a template to build the other strand, the complementary strand, and now you will end up with 2 double-stranded molecules. So here, 5 prime, 3 prime, T A C G, and then down here, 3 prime, 5 prime, A T T G C. Next thing, the cell's cytoplasm separates, pinches off, and each cell takes with it one of the 2 double strands that have been made. Notice that what the replication gives us is 2 double-stranded molecules. Each of the double-stranded molecules, each of them is going to get an old strand and a new strand. New strand, old strand. Okay. So there's the synthesis of each strand. And the fact that each of the new cells will get a parental strand and a new strand and parental strand. We describe this replication as being semi-conservative. So the term semi-conservative is describing DNA replication, which gives us 2 double-stranded molecules, each with a new strand, each with an old strand. So you should have the meaning of antiparallel DNA strands, complementary DNA strands, semi-conservative DNA replication. And again, now given one strand, you should be able to get the new strand. So just make another one so we don't do that one. So let's say here this is 5 prime, and this is C, C, G, T, fill in everything else to create the other strand. So here, if that's prime 5, that will be 3 over here, and the new strand will be 5, 3, G, G, C, A. Okay. DNA replication requires enzymes. The main enzyme, the one without which DNA replication cannot happen at all, is DNA polymerase. DNA polymerase is described as the anabolic enzyme. This is the enzyme that actually replicates DNA. Now, DNA polymerase has two constraints, two things that it cannot do, or that it has to do, I guess I should say. It has to replicate DNA moving always in the 5' to 3' direction. And I'll show you what that means here in just a second. Number one. Number two, it cannot start replication. It can only continue replication. So those two constraints have to be met, have to be dealt with. Okay, so one of the constraints is that it can only put the nucleotides in the 5' to 3' direction, which means that it is going to take one nucleotide and attach the 5' end, the phosphate end of one, to the 3' end of the other. And then the next one nucleotide will be 5', 3', 5' to 3', and continue in that manner. So what it cannot do is start with a 3'. Okay, this is not. All right. You cannot start with a 3'. Let's see what we can do this. So it will be in this direction. Okay, you cannot start with this guy. And attach 3' to 5'. It cannot do these. Okay. You can only attach the 5' to the 3'. So it will proceed 5', 3', 5', 3', 5', 3'. That's a constraint, and you'll see why. Okay. Okay, here's explaining the constraint. Let me see if we can do these. All right, well. Okay, so if you have here the origin of replication, and the way DNA is going to replicate is bidirectionally, meaning you have one parental strand and you have two parental strands, but when you look from the OR, you have a replication that could be going using this side of the parental strand, this side of this parental strand, this side of this parental strand, and this side of this parental strand. And each one of these sides is going to be used for replication. So that's what we call it bidirectional replication, because we're replicating on both sides of the OR, of the original replication. So if we take, and even though this is a circle, it's going to replicate from the OR on either direction, having two templates on each direction. So from the OR, if we call this one, if this happens to be the 5' end, right here, and this is the 3' end. That means that on the other side, this is 3' and 5'. Next to the 5', the next nucleotide will be a 3'. So on the other side of the OR, we'll start with a 3' and with a 5', and on the opposite, 5' to 3'. Okay, so remember, DNA polymerase can only work 5' to 3'. It can only put nucleotides in those directions. Okay, so when replicating this strand right here, I'm going to make it thicker. We're going to replicate this strand right here. DNA polymerase cannot start at the 5' and begin putting a nucleotide, and then just go ahead and do something here. Let me erase these for now. Okay. Okay, it cannot start here and move in this direction, because if it were to do that, it would be going 3', 5'. Remember, it has to be anti-parallel to the template. It will be 3', 5'. It cannot do that. It has to go in the opposite direction. So when DNA polymerase replicates that strand, it has to move towards the ER. So it's putting the nucleotides towards the OR. The other strand has no problem because this is 3'. It can move away from the OR without any problem. So the molecule that is moving towards the OR is the one that is used in the 5' to 3' parental template. The one that's moving away from the OR is the one that's used in the 3' to the 5' template. Okay, so here, 3' to 5', it can move away from the OR. 5' to 3', it has to move towards the OR in order to go 5 to 3. So that's one of the constraints that it has, that it cannot just move in any direction. The second constraint is that it cannot start DNA replication, it can continue it. Okay, so, and this is true of all DNA polymerases. They cannot start replication. Okay, so they're going to have to rely on another molecule, another enzyme called a primase to start replication. The problem with the primase, again, all DNA polymerases do not have the ability to start replication, which means that a primase cannot use DNA, because if it did, it would not be able to start replication. So primases are actually RNA polymerases. But we don't call it an RNA polymerase, we call it a primase. It uses RNA nucleotides. It is going to synthesize about 10 of those. We call them the primer. You can see here, each one of these DNA molecules, it starts out with the primer, the primase, creating the primer, 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 and then from the primer, DNA polymerase can take off. The problem is that now we have a little piece of RNA that has to be removed and changed to DNA. So the cell is going to have to do that before DNA replication is finished. So it becomes a very complicated. Is that a safeguard against crazy growth, the fact that polymerases can start replication without primases? It is definitely a constraint. On the one hand, absolutely is a constraint. And DNA polymerase is a later, evolutionarily speaking, is a later molecule. RNA polymerase is a much more ancient molecule than DNA polymerase. So it is a constraint, absolutely, so that the cell can control the polymerase. Absolutely. Okay, so there is the two strands that they're being replicated. Notice that again, just back to the same thing, the 5' to 3' as the parental strand, the new strand has to move towards the OR, 3' to 5' as the template, DNA polymerase can move away from the OR. And same thing on the other side, but opposites. So, okay. So at this point, you should know what is a DNA polymerase, what is a primase, what is a primer. Notice the problem that we have here. If we're replicating towards the OR, comes a point in which we hit the OR, we cannot go any further. So that DNA polymerase has to jump back and start again a little bit further back. Primase makes the primer, DNA polymerase takes off, it starts copying, and all of a sudden bumps against the previous strand. So now DNA polymerase has to jump back and start over again. The primase makes the primer, and then DNA polymerase takes off from there, keeps going, and then bumps against the previous strand, and then it has to keep repeating itself. So it has to keep going back and forward, back and forward. So the strand that's being replicated in this manner is called the lagging strand, and is being replicated into little pieces called Okazaki fragments that the cell is going to have to join later on. The other strand is fine because it's moving away from the OR. It can just begin and then keep going without stopping. Same thing up here. 5' to 3' moving away from the OR just starts replicating and doesn't have to stop. 5' to 3' towards the OR has to be replicated in little fragments. So now the cell has to go back, remove primers, fill them in with DNA, and then join the fragments together. So it's a lot of work, this DNA replication business. All right, next thing that you need to know. So at this point, what do you need to know? What you need to know, what do we mean by directional replication? What is the anabolic enzyme of DNA replication, DNA polymerase? What are the two constraints of the enzyme? What is a primase? What is a primer? And now we're going to talk about other enzymes that are involved in replication. Oh, and the location of what this happens. The location of prokaryotic replication, anywhere where DNA is found, in this case in the cytoplasm. In eukaryotes, it will be in the nucleus and the mitochondria. All right, other enzymes, helicases are enzymes that are going to unwind DNA, so unwinded and separated. The two strands that are separated are being kept apart by stabilizing proteins, which are these yellowish proteins right here. Then DNA polymerase can begin binding and begin first the primase, and then the DNA polymerase can begin replicating. I have no idea what that is. Let's see. Okay, the problem with this picture is that it is giving me the OR is somewhere in here, and it's only giving me one strand. I'm not seeing what's happening at the other side of the OR, but that's okay. So this is the 5' to 3' moving away from the OR. There's the primer. DNA polymerase can move on without any problem. The other strand is the lagging strand that it's moving towards the OR in that direction. The helicases are unwinding DNA as DNA is being replicated. Stabilizing proteins keep the two strands separated. Okay. Just want to make sure I didn't look at those. Another enzyme. Okay, so we have the primase, we have the DNA polymerase, we have the Okazaki fragments. You should know what those are. So here you have the primer being removed and DNA polymerase filling in the gap. Okay, another enzyme that is going to be needed are the ligases. which are going to join the Okazaki fragments. The primers will be removed by an enzyme called RNase H, and then DNA polymerase will fill in the gaps. So now we're up to what enzymes needed? The main enzyme, DNA polymerase. Without it, you cannot have DNA replication. Other enzymes, helicases, ligases, primases, RNase H. Okay? And then stabilizing proteins. Stabilizing proteins. Okay? Definitely know everything that's involved in DNA replication and the function. Okay, so again, this I like this picture better because it's giving you the OR and both, and then what's happening on either side. So here you have the continuously replicating strand. Yes, they should be. But are any of those peculiar to prokaryotes? Yeah, these are the same enzymes in prokaryotes and eukaryotes. So you find exactly the same enzymes in both eukaryotes and prokaryotes. So far, none of these enzymes are any different. Yeah, absolutely. So that's the beauty of it. This is the lagging strand on the other end, leading strand, lagging strand. So it gives you further the picture of how it's happening. The replication fork are these little forks being made as DNA is being replicated. Okay. All right. Now, after DNA is replicated, and this happens in both bacteria and humans, we saw it first in bacteria, obviously, and then it turns out human cells do something similar. This is something called methylation of DNA. And methylation is putting a methyl group in certain places of the DNA. A methyl group is simply a carbon with 3 hydrogens attached. That's a methyl. So what methylation does, and so what the cell is going to do is it's going to add this little methyl group in some of the bases. And it may appear random, but it's not random. It is a little secret code, quote unquote, that the cell has to be able to recognize its own DNA. So part of the purpose of methylation is to protect itself from foreign DNA. Foreign DNA is not going to be methylated the way the cell has methylated its DNA. It's like a branding of the DNA, saying this is mine. Viral DNA, for example, will not have that branding, and the cell will be able to recognize the DNA as foreign, and it will be able to attack it. It is also places where the cell uses to control gene expression. It may say, okay, this is a gene that is going to be turned on or turned off. Yes, in eukaryotes, methylation is what happens with epigenetics. In other words, it's something that happens by exposure to the environment, by different chemicals in the environment or different environmental factors. Not so much so in bacteria. Yeah. So yes, yeah, exactly. So it's not so much in bacteria. Now, bacteria is more a branding. Places where DNA is going to be marked for, let's say this is a gene that we want to, that the cell wants to express, or not. The place, for example, the ORs are methylated, so the cell knows this is an OR, or places where DNA has to be repaired, for example. Okay, so these are markings that the cell is going to use. So bacteria, yes, on purpose in bacteria. Yes, it is. It is not random. Yes, the methylation is on purpose. Absolutely. And in the sense, in the sense of it has a specific reason. It's not random. It's not random at all. Okay, so yes, it's not random at all. Whereas in humans, it tends to be more random. In bacteria, it's not random. This is a bit of a comparison between DNA replication in prokaryotes versus eukaryotes. So E. coli, the size of the DNA is obviously much smaller. This is 4.7 million nucleotides versus 150 million nucleotides in humans, nucleotide pairs. The replication rate in prokaryotes replicate at 1,000 nucleotide base pairs per second. Humans, 50 nucleotide base pairs per second. So humans not only have much more DNA, the replication proceeds much slower. We know from generation time that a cell takes about 20, 30, 40 minutes to replicate. So that's how long it takes it to copy its DNA. In humans, it will take months to replicate the DNA if there was only one OR. Human DNA has more than one OR, so it ends up taking about an hour. So we make up for it by having more origins of replication. Nonetheless, the replication of prokaryotes proceeds much, much faster than that of eukaryotes. So we talked about DNA polymerase having two constraints. It cannot start replication. It can only continue it. That's one constraint. It can only move 5' to 3'. However, it has one huge advantage, which is unique to DNA polymerase. It can proofread itself. It can proofread its work so that if it makes a mistake and a G happens to attach to an A, and yes, a G and an A could make bonds. It would be they're very unstable bonds, but they could potentially bond. DNA polymerase can catch that mistake, remove the nucleotide that shouldn't be there, and replace it with the good nucleotide. So that is a tremendous advantage that RNA polymerase doesn't have, for example. So DNA polymerase has the ability to check its work so that a mistake is made rarely. It's an extremely rare event when the proofreadability when DNA misses a mistake. Okay, it occurs in the rate of one mistake per 10 to the 9th, which would be what, a billion, I guess. So 1 in a billion. So it would put a billion nucleotides correctly, and if it doesn't put them correctly, it catches the mistake and it fixes it, and maybe one for a mistake that was not caught. Okay. What this tells us, we're going to see these in chapter 8, is that mutations due to DNA polymerase mistakes are rare, incredibly rare. It just doesn't happen because DNA polymerase can check its mistakes. Okay. Mistakes that are missed are called spontaneous mutations, and they are extremely rare. All right, so you should know you should be able to explain semi-conservative replication, bidirectional replication, lagging strands, Okazaki fragments, origin of replication, replication fork, and then throw into the mixture all the enzymes that are involved in DNA replication. So DNA polymerase, helicases, RNA-H, ligases, primase, and stabilizing proteins. Okay. Given the double-stranded DNA sequence below, determine which strand is the template for the leading strand and which is the strand, the template for the lagging strand. Okay, so we have the OR right here. Okay, and then we're not looking at what's happening on the other side of the OR, just on one side of the OR. We have a 5', 3', and then 3'. Make these a little bit bigger. Okay. So we 5 prime, 3 prime, 3 prime, 5 prime. So as far as the new strand, up here, it will be 3 to 5. And down here will be 5 to 3. So 5 to 3, go in that direction, uninterrupted. 5 to 3 will have to go in that direction, interrupted. So these will be lagging. This will be continuous. Okay. Questions about DNA replication? I'm going to go ahead and push it transcription just because it is not that bad. And after transcription, we'll go ahead and stop there. We'll not go through translation. Well, yeah, it's not as bad. It really isn't. So let's go ahead and push it through. What's this? Okay. All right. Okay, so transcription has to do with the function of DNA, which is to code for the synthesis of proteins. Okay, so the first thing to know is, where does this happen? Okay, so where does transcription happen and why does it happen? What's the purpose of transcription? This is the deal. If you look at the DNA as a recipe book, okay, that has the recipes for all of the proteins the cell will ever need. The place where the proteins are going to be made are going to be in the ribosomes. The ribosomes are the site, the organelle that is in charge of protein synthesis. Okay, so in the eukaryotic cell, the need for transcription is very obvious because you have the recipe book in the nucleus. You have the kitchen where you're going to cook the recipes in the cytoplasm. So now you have a problem. You have two different rooms. So you have two choices. You either bring the ribosomes to the nucleus to use the recipe book to cook the proteins and make them, or what you could do is, instead of taking these, there's no way you can take these entire encyclopedia of recipes out of the nucleus. There's just no practical way to do that. The next thing will be, let's just copy the recipe. So copy the recipe that you want, that the cell wants. Now that small copied recipe can be taken out of the nucleus into the ribosomes and used to make the protein. So that's exactly what the cell is going to do. It's going to make a copy of the recipe. That's what we're going to call transcription. And that copy of the recipe will be sent out to the location of protein synthesis, which are the ribosomes, and the protein will be made using the recipe. So the reason for transcription is very obvious in the prokaryotic cell, in the eukaryotic cell. In the prokaryotic cell, it's not as obvious because you can argue both the recipe and the kitchen are in the same location. They're both in the cytoplasm. So there's no need to be doing that except to be, copying the recipe, except that what you have is a tremendous mesh of DNA. And somewhere in there is the recipe that you want. So instead of having to find that recipe and trying to bring the ribosomes to the recipe, again, it is more practical to begin copying the recipe and use the copy to make the proteins. So transcription, which is the copying of the recipe of the protein instructions, also happens in prokaryotes, even though the need for that is not as immediately as obvious. Okay, so transcription is the process that makes an RNA copy of a gene. Okay, so why RNA? Why not DNA? Well, DNA is a more precious molecule, is a more stable molecule. It will be the equivalent of writing the recipe in a nice piece of paper with a nice pen. If you're jotting down a recipe quickly, you may just get a pencil and begin jotting down the recipe. RNA is a pencil. It's cheap, abundant, not very stable. You can, get rid of it. And so it is expendable. DNA is not expendable. RNA is expendable. So that's why the cell will use RNA and not DNA to make the copy. Okay, so the process is called transcription, the copying of the gene. The actual making of the protein is going to be called translation. Or expendable means that in this context, is that it is energetically speaking and in terms of raw material, RNA is easier to get rid of and make new. So it's cheaper, energetically speaking, and raw material speaking. It is easier for the cell to make more RNA, than it is DNA. DNA is a lot more expensive to the cell, and expensive in a cell means takes energy, takes raw products that are hard to make. RNA is everywhere. You have RNases, enzymes that destroy RNA all over your fingers. I mean, enzymes that destroy DNA are everywhere. RNA is everywhere. It's a very common molecule in the living world. Let's see, then transcribe to RNA. Okay, so we transcribe DNA to RNA, and then the transcription, the copy, is then translated to form the polypeptides that makes the proteins. But we're going to talk about transcription first. We'll talk about translation later. The three products of transcription are going to be the copy of the recipe, which we call messenger RNA, and then two other RNA molecules that are going to be involved in protein synthesis, the ribosomal RNA and the transfer RNA. So both of these are going to be made through the process of transcription, plus the copy of the gene, which is the messenger RNA. Definitely know the three products of transcription. Definitely know that out of the three products of transcription, the only one that is translated is going to be the messenger RNA. The other two are going to remain as RNA molecules. So one is the kitchen, the other one is like a little helper that is going to be involved in making proteins. This is what tRNA looks like. This is what ribosomal RNA looks like. So T stands for transfer RNA. Again, for location. Notice that we always go for, look at the location. Where is it happening in the eukaryotic versus the prokaryotic cell? The answer is simple. Wherever DNA is. Wherever the recipe book is, that's where we're going to have transcription copying the recipes. So in the case of prokaryotes, in the nucleoid of the cytoplasm, in the case of prokaryotes, in the nucleus and the mitochondria and chloroplasts of eukaryotes. Products of transcription, again, you should know those. Okay, the steps are straightforward. We start, we keep going, we finish. So initiation, elongation, termination. Enzymes involved, simple. Basically one enzyme, RNA polymerase. So obviously this is a much simpler process than DNA replication. RNA polymerase has one constraint. It has to synthesize in the 5' to 3'. It's a constraint that all polymerases have, regardless of whether they are RNA or DNA polymerases. They always have to move 5' to 3. Doesn't need a primer like DNA polymerase does. However, it does not have proofreading ability. So it's not a sophisticated enzyme. It's a very simple enzyme. It copies If it makes a mistake, couldn't care less, keeps going. Okay. So the start of transcription begins at a place in the gene called the promoter region. It is not a place where the recipe is located, but is in front of the recipe, and it's just the start place. So this is where you're going to start copying. It's the place where RNA polymerase binds to the promoter region, so it's going to bind to this region, and from there, RNA polymerase will begin to unwind and separate the strands. So notice that RNA polymerase can do that, the work of helicases and itself. In the case of prokaryotes, so the sigma factor is prokaryotes only. It's a little piece of the RNA polymerase that is helpful in recognizing the promoter and unzipping the DNA. And then as soon as the RNA polymerase is attached, DNA is unzipped, sigma factor falls off. So here's the promoter. The promoter region has a lot of A's and T's. It's called the Tata box. Because remember that T's and A's are attached to each other by 2 hydrogen bonds instead of 3. So they're going to be easier to unzip. So RNA polymerase attaches to the promoter region. That's what we call the recognition site. The sigma factor falls off as soon as the 2 strands are separated, and RNA polymerase can begin copying. To make the copy of the recipe, all you need is 1 copy. You don't need both copies. So RNA strand is going to be a single-stranded molecule that will be copied. Because it can only go 5' to 3', that means that the strand that is going to be transcribed is going to be the 3' to 5'. So in this example, this top strand is the 3' to 5'. That's the one that's being transcribed. It's the one on top, right there. The other one is not being transcribed. Okay, so you can see here the nucleotides put in place. Remember that instead of T's, and uracil is going to be used. So what will be happening, because the cell needs many copies of the recipe, there will be many RNA polymerases, one after the other, copying the recipe. So the cell is not going to make 1 molecule of messenger RNA. It's going to make copies of the cell, of the recipe. Termination is can be done on two mechanisms. One of them is by forming what's called a hairpin loop, which is essentially a loop of the RNA that is going to pull away from the DNA. Or there will be a protein that is going to terminate transcription. So as soon as the cell reaches the end of the DNA gene, a hairpin loop or a protein is going to stop transcription and pull the RNA polymerase out of the way. Prokaryotic genes are polycistronic. This is what it means. Remember that prokaryotic DNA is smaller, so there is a constraint on size. There's fewer genes. There's less DNA. So if there are a series of genes that code for proteins that are involved in the same process, then it makes sense to have 1 promoter for every one of those genes instead of having a promoter for every single gene. In other words, if gene 1, gene 2, and gene 3 code for proteins that are involved in the same pathway, let's say gene 1 is the first enzyme in the pathway, gene 2 is the second enzyme in the pathway, gene 3 is the third enzyme on the pathway, why waste DNA creating a promoter in front of each of those genes. When the cell needs gene 1, it's also going to need gene 2 and gene 3. Let's just have one promoter in front of all 3 genes, and the transcription process will make one long messenger RNA that could be translated into 3 different polypeptides. That's what we call a polycistronic messenger RNA. This works because the cell, when it's making the recipes, there is the start of a recipe always begins with the same 3 nucleotides, AUG. The end of the recipe, there are 3 nucleotides that could signal the end of the recipe. UAA, so that says stop. That's the end of gene 1 recipe. AUG, okay, that's the start of the next gene recipe. UAG also means stop. Okay, that's the end of gene 2 recipe. AUG, start of gene 3 recipe. UAA, again, is another stop signal. So because there are start and stop signals, the cell knows when each of the 3 recipes begin and end, and it knows, polypeptide 1, polypeptide 2, and 3 are synthesized. So polycistronic messenger RNA is a time saver, space saver for the prokaryotic cell. So you should know what we mean by polycistronic messenger RNA. All right. We're going to end by looking at eukaryotic transcription. Eukaryotic transcription is a lot more complex than prokaryotic transcription. Part of the reason why is because it involves more than one step. The recipe that is copied, or what is being copied, has to be modified to create the final recipe. The process is called post-transcriptional modification. And it actually makes a lot of sense. It's a way for cells to save resources. Because if prokaryotic DNA is big, eukaryotic DNA is 1,000 times bigger. And out of that incredible mesh of genes, the cell may only be using maybe a few of those, maybe 10% of those genes are being used all the time. So what if you could have a piece of DNA that, when copied, the copy could be modified to create different proteins. So now you have one recipe, and that recipe, you could take away some of the ingredients and have one dish, or take away other ingredients and have another dish. And you can modify the end protein by just modifying the pieces of the recipe. That would be a tremendous time-saving, space-saving, energy-saving process. So that is exactly what's going to happen in eukaryotes. So here's a gene, or let's call it a coding region instead of a gene. Okay. So from that coding region, there are pieces of that coding region that are going to be trash, that are going to be not part of the recipe. We're going to call those introns. The part of the recipe that is going to be used are going to be called exons. Okay? So RNA polymerase is going to copy the entire recipe, everything. So here's the pre-messenger RNA, the copy of everything. Then specialized enzymes are going to come along, and they're going to recognize the trash regions and cut them away. Okay, so we don't want that one, intron 1, intron 2, and those 3 pieces are going out, and the pieces that are part of the recipe are going to be joined in. And now we have the actual messenger RNA. Okay, so the actual messenger RNA will be taken out, and let's say protein A is made from it. Okay, well, let's take the same recipe, but now let's change the components. And now we're going to say these green parts are going to be kept, and the blue parts are going to be taken away. So when that pre-messenger RNA is processed, we're going to end up with the green parts coming together. Am I right? Yeah, the green parts of the entrance now, or they're going to be the exons. So the green part is going to be the messenger RNA. The red parts are going to be destroyed, cut away, and now we have a different recipe that will give us protein B. And you can see how you may end up with infinite combinations with just one coding region. You could modify and get many different versions of the same, I guess, protein, if you will, okay, or different proteins from these basic components. That is a tremendous saving for the cell. Okay, so this is it's very complicated, obviously. It adds a level of complication to transcription in eukaryotic cells. So eukaryotic cells go through post-transcriptional modification. And you can see why transcription was first figured out with bacteria, because bacteria is straightforward. What you see is what you get. With eukaryotes, what you see may not be what you get. So it just puts it makes working with eukaryotic organisms a lot more difficult. Okay. So compare and contrast eukaryotic transcription and prokaryotic transcription. Okay, we just talked about RNA polymerases, one having a sigma factor in the prokaryotes, no sigma factor in eukaryotes, post-transcriptional modification in eukaryotes, no post-transcriptional modification in prokaryotes, polycistronic messenger RNA in prokaryotes, none of that in eukaryotes. Would prokaryotic RNA polymerase be a good target for antibiotics? Okay, how close are the prokaryotic and the eukaryotic RNA polymerase? They're actually fairly close, enough to where antibiotics that target RNA polymerases are typically more toxic than other antibiotics. So is it a good target? It's not a perfect target, but it is used nonetheless. Put into the picture the fact that RNA polymerase in mitochondria is going to be very similar to the RNA polymerase in prokaryotes. And then that makes those antibiotics even worse. Okay, so post-transcriptional modification, introns out, exons in, and then advantages. We kind of talked about that. Okay, now you should be able to transcribe. So given the following DNA sequence, choose the appropriate DNA strand to transcribe. And notice that we're telling you the transcription is proceeding in this direction, which means the promoter region is in this direction. So the promoter, I'm going to put P for promoter, is over here. Okay, so the promoter region is on the left, and I know that RNA polymerase has to copy or has to move 5' to 3'. That means that it has to transcribe the 3' to 5' strand. So that the messenger RNA is going to be 5', 3' is going to be messenger RNA. A, U, G, U, C, C, G. We're in a room here. CCUAA, CCUAA, okay, which is very different. If I make a mistake and I transcribe the one below, we're going to end up with a completely different sequence. So be prepared to do that. Be prepared to, if you're given double-stranded DNA and you're told that the promoter region is, let's say, on this side, you should know which of the two strands should be transcribed and then make the transcription. create the messenger RNA. Okay. All right, questions? Okay, so we have about 30 slides left. So we should be able to finish transcription next time. And chapter 8 is not this long. Yes, so we're meeting on Thursday at 8:30. Correct. So the next time we meet is Thursday at 8:30 to finish chapter 7. And then start chapter 8. We'll definitely start chapter 8. Yep. Okay. Okay. Okay, so you're going to have lots of questions. That's fine. Yeah, no problem. So next time we'll deal with questions. Very good. So that's good. So we'll definitely address those next time. So you need to digest all these now.